So here is the hormones component to the exam 5. I included this slide in both PowerPoints for hormones and cell signaling just in case people watch one or the other so they can see it. So Dr. Correa swears by this exam master because he said one student told him that all they did was work these questions and they did pretty well on the board. I suggested it last year and my M1 year and got kind of mixed results or mixed feedback about how useful this actually was. So if you're interested, maybe give it a try. Maybe it'll be worth it. Wish I could say something for sure. So the Rapid Review Biochemistry book, mentioned this at the beginning of the year. It's really good. There won't be anything on the board that's not in this book. So you've got some time before you actually take your board exam to order if you're interested. And then here's a link to a PDF that NBME puts out for the subject exams. So this includes information about all the subject exams from between M1 and M3 year. And it kind of gives you a breakdown of the topics they like to test on, what percentage of questions from each topic will show up on the exams, and they give you like 10 or 15 or so practice questions within this PDF. It's kind of helpful, and at least gives you kind of an idea of how you're going to be tested. So the hormonal cascade system is very similar to the cell signaling cascade system in that you're going to start with some sort of stimulus, and then eventually you're going to get down to your final product, and it's going to be pyramid. So you're going to have very little stimulus that can result in large amounts of actual product being generated. So here we have an environmental signal, and it's going to stimulate our central nervous system, which is going to, in this example, going to stimulate the limbic system, which you'll learn about in neuroanatomy, maybe in phys, so don't worry too much if you don't know what that is. But that'll activate the hypothalamus. Then the hypothalamus will send signals to the anterior pituitary, and the anterior pituitary will then send signals to a target gland, and then you'll get this gland will eventually release the hormone that's going to allow your body to respond to the initial stimulus. So it's important here to look at and see that the hypothalamus will release nanograms, anterior pituitary will release micrograms, and the target gland will release milligrams. So you increase the amount of secreted product as you get closer to the end of the cascade. Also want to point out that's very important here is that the half-life increases throughout the cascade. So this product here, there's going to be very little of it, very short half-life. This will be kind of in the middle, medium amount, sort of medium half-life. And there's going to be a lot of this and it's going to have a long half-life. So there's a lot of it that's going to stick around for a while. So this is an anatomical diagram of what we're going to be talking about for most of this, is that we have the hypothalamus here, and then you have the pituitary stalk, and then the pituitary gland here. So the anterior pituitary, posterior pituitary, and you'll learn more about this again in neuroanatomy. So the important things for this that'll be testable is that from the hypothalamus, the neurons go from the nucleus, and when they go into the posterior pituitary, they release the hormones into the bloodstream directly into the posterior pituitary. However, with the anterior pituitary, the neurons release the hormones upstream in this is called the hypophyseal region, and they release this up here, and then the hormones have to travel down the pituitary stalk into the anterior pituitary to reach the target cells. So there's a little bit of a difference in how signals are received by the anterior pituitary and how signals are received by the posterior pituitary, and that's a very testable concept. So this is probably the most high yield slide out of all of the hormones content. So I'll just kind of walk through it. Last year, the students complained that I didn't really work through it because I was just like, if you can memorize this chart and understand how this chart flows, then you'll probably do pretty good on the test, or at least for the hormone questions. So I'll just kind of walk through it real quick. and maybe it'll be helpful. So we're going to have some sort of signal that's going to activate or act on the hypothalamus, and then this is the anterior pituitary here. All of these guys here are anterior pituitary. So you're going to act on the anterior pituitary, and the anterior pituitary is going to release several different hormones that's going to result in some sort of biologic response. So we'll start here. So if the hypothalamus releases growth hormone releasing hormone, then you're going to activate the secretion of growth hormone, and that'll act on all the tissue, a lot of tissues in your body, to secrete insulin-like growth factors, which are going to stimulate growth, or they'll act on muscle, which is going to increase protein synthesis and decrease glucose uptake. And this occurs by insulin resistance. I don't know if that's going to show up on this test, but that's a concept that'll come up repeatedly, is that growth hormone and cortisol both cause insulin resistance, and that's why you get a reduction in glucose uptake. They'll act on adipose tissue to increase lipolysis, so freeing up fatty acids for energy. And then they'll also work on the liver to increase gluconeogenesis as well as glycogen synthesis. So this is very similar to cortisol in that you have sort of a fed and fast system activated at once. It does seem kind of paradoxical, but it has to do with the buildup of gluconeogenic substrates. So gluconeogenesis is kind of going as fast as it can. Those substrate build up, and then they kind of get forced down the glycogen synthesis pathway. I know it's kind of paradoxical, but that is in fact what happens. So this whole system here is inhibited by somatostatin. So the next one we're going to look at is going to be thyroid hormone. So the hypothalamus is going to release thyroid releasing hormone, which is going to act on the anterior pituitary. The anterior pituitary is then going to secrete thyroid stimulating hormone, which is going to act on the thyroid gland itself. This will cause the thyroid to secrete the T3 and T4, and this increases protein synthesis and basic metabolic rate. And then we have corticotropin releasing hormone, which acts on the anterior pituitary to secrete this POMC, which is a long peptide that gets chopped up into a lot of pieces, and each of those pieces has some sort of biological function, which you'll learn about in phys. But in terms of this, you need to know that it gets chopped into a small component called ACTH. ACTH goes to the adrenal cortex, stimulates the secretion of cortisol, and then it causes anti-inflammatory response and immune suppression. And probably some of the biggest things about cortisol is right there. In the muscle, you're going to get increased protein breakdown. So this is in juxtaposition to growth hormone over here, because in growth hormone you're making protein, but cortisol causes protein breakdown. You're going to get lipolysis from adipose tissue, and from the liver you're going to get gluconeogenesis and glycogen synthesis, just like with growth hormone. So sometimes you'll see a question and you're not really sure if it's going to be cortisol or if it's going to be growth hormone that's causing it. So what you need to do is you need to look at, are you making proteins or are you breaking proteins down? If they don't give you the anti-inflammatory and immune suppression information. So then the next part is going to be the stimulation of sort of like the gonad hormones with FSH and LH. So we have gonadotropin releasing hormone, which is just going to stimulate the anterior pituitary to release follicular stimulating hormone and luteinizing hormone. And it's important to know which, if it's testes or ovaries that these are acting on. So in testes, you're going to secrete. The FSH is going to act on the Sertoli cells. This is going to cause the Sertoli cells to secrete APP, which is going to lead to the biogenesis of sperm. You guys have probably talked about this pathway a little bit in histo at this point, I believe. And then LH in the testes is going to act on the Leydig cells to generate testosterone, which also leads to the development of sperm. FSH with the ovary is going to lead to the development of a follicle within the ovary. It's going to lead to the generation of estrogen. And LH also acts on the ovary to either induce ovulation or sustain the corpus luteum, which then secretes progesterone and E2. And this occurs early in development when the placenta is unable to produce these hormones itself. And then this whole process is inhibited by gonadotropin releasing inhibitory hormone. So whenever you're taking the test and you're in a hurry and you're anxious, don't get GnRH mixed up for GHRH, or this somatostatin is also sometimes called GHIH. I don't know if Dr. Romero uses that terminology, so just don't get these confused. GN here, gonadotropin, just a G, or GH is growth hormone. And then, so the last one is going to be the stimulation or the secretion of prolactin. So we're going to have prolactin releasing hormone. It's going to stimulate this. It's going to affect the mammary gland, and it's going to lead to the synthesis of milk proteins. And dopamine inhibits this process. So this one's a little bit tricky because what we do is we initially secrete our basal sort of process is that we secrete dopamine. So by default, this is turned off. And when we need to generate milk, the response is to inhibit dopamine, which then allows prolactin to have its effect. Okay, so the next part is going to be the posterior pituitary, and it's easy. The only two hormones it secretes are going to be oxytocin and vasopressin. So vasopressin, is going to become active in response to low blood volume or high salt. So vasopressin's got a lot of different names. Vasopressin and antidiuretic hormone, or ADH, are going to be the most common ones you're going to see. And the idea is that you're going to retain water. So if you have low blood volume, you're going to retain water. Or if you have a really high salt concentration in your blood or sodium concentration, you're going to try to dilute it out by retaining water. And this acts on the kidney, and as I said, stimulates water resorption. The other hormone that the posterior pituitary secretes is oxytocin. And this will be secreted by mechanical stimulation of the cervix, or breastfeeding and mechanical stimulation of the areola. So this will cause the posterior pituitary to release oxytocin, which causes the uterus to contract or the mammary glands to eject milk. And this is a very important distinction between oxytocin and prolactin. In addition to prolactin being anterior, oxytocin being posterior, is that oxytocin is actually milk letdown, and that prolactin is only the synthesis of milk proteins. So if you have functional prolactin and functional oxytocin, you'll be making plenty of milk, but you won't be able to eject it. And vice versa, if you have plenty of oxytocin, you can eject milk, but if you're not making prolactin, then you have no milk to eject. So this is just a pretty simple slide, and one that he'll ask a lot of questions about, and that we know very little about what actually causes hypertension. So this is probably, maybe 15% or so of what we know about hypertension. So there's a lot about hypertension that we don't know. So the way he'll ask a question is he'll give like a list, and it'll be like, which of the following is most likely to cause hypertension, or something along those lines, and it'll be something from this list that's causing the hypertension, because the rest of it we simply don't know. And what we don't know is called essential hypertension, which is a fancy word for we don't know, it's magic. Questions from this slide have come up a little bit. So we have vasopressin here, also ADH, or arginine vasopressin, and then oxytocin. So both of these are kind of interesting because they come from this long precursor peptide that'll get chopped up, and then you'll get your active vasopressin. And the same with oxytocin. You make this long peptide, and then it is cleaved. The actual peptide is cleaved to generate product. So the way the question is, it'll something along the lines of like, which of the following hormones is generated by cleavage of a protein into smaller components, or something along those lines, and then it'll be vasopressin and oxytocin is what they're getting at. All right, so none of these hormones are super complicated, but you just got to spend a little bit of time to make sure you get them straight. So we'll talk about ADH, or arginine vasopressin, antidiuretic hormone. Vasopressin is all the same thing. So it's secreted by the posterior pituitary, and it acts in the kidney. So here's a diagram of a nephron. You've got the proximal tubule, loop of Henle, distal convoluted duct, collecting tubule, etc. But what's important is that the vasopressin, ADH, whatever you want to call it, acts in the collecting duct, and it causes the stimulation, or let me back up a little bit. So ADH is going to act on a G-coupled protein receptor. This is going to stimulate the cyclic AMP cascade, so cyclic AMP PKA pathway, and this is going to lead to the transition of a vesicle containing aquaporin 2 to the luminal surface. So this is the inside of the collecting duct, and this is like interstitial tissue over here. So we're reabsorbing this way. So we're going to put these aquaporin channels here on the luminal side, and this allows water free entry into the cells, and then it is pumped back out this way. So this is how we dilute our urine, maintain blood pressure by reabsorbing water. So some of these things over here I mentioned, so cyclic AMP, that it acts on this through the cyclic AMP pathway is important. That it's a peptide hormone, so not a cholesterol or steroid-based hormone. Aquaporin 2 is how it resorbs water. Those are the high points. All right, so for diseases related to ADH or vasopressin, there's a couple of diseases, big diseases that will show up. There's a little bit more, or like subtypes of each of these, but don't get too crazy with it. He doesn't want you to know those. You'll learn more in Phys and Path when the time comes. So the first one is diabetes insipidus. So it's either a hormone deficiency, you're not producing enough ADH, or hormone resistance. Your kidneys just can't respond to ADH. So you're not able to recruit these aquaporin 2s to the surface. So you have a really high water content in your urine. So you're going to have very dilute urine. You're going to have troubles maintaining your blood pressure because you can't reabsorb water. And you're going to have high sodium in your blood because, again, you can't dilute out your plasma by resorbing water. So that's kind of what I just said here. Urine osmolality is decreased, and that's just because you're really diluting it out because you're letting all the water go through the collecting duct and not resorbing any of it. So there's a couple of things that can cause this. You can have trauma to the pituitary stalk, and you're not able to release ADH, or inflammation, infection. Basically anything that could manually compress or disrupt the posterior pituitary. There's a few instances of genetic mutations, but the decreased secretion and the decreased renal responsiveness are the big ones because this will be neurogenic, and this will be what's called central. You don't need to know that yet, but these are the two big ones. So decreased renal responsiveness to antidiuretic hormone, and the treatment is to basically give them the hormone back, and the hormone that they make in the farm industry is called desmopressin. This is not naturally occurring. This is the made in the lab one. All right, so after diabetes insipidus, the next major disease that we want to talk about with the ADH is going to be syndrome of inappropriate ADH. So this is basically going to be too much. So diabetes insipidus is not enough action here in the collecting duct, and SIDH is too much action here in the collecting duct. So here we're going to have a very small volume of highly concentrated urine because we're going to have tons of these receptors come into the luminal surface. We're going to be resorbing a ton of water. And because we're resorbing a ton of water, we're diluting out our plasma, and we're going to have a low sodium concentration. So this you'll see can either be caused by AVP secreting tumors. So if you have a posterior pituitary tumor, it can either be from the cell that secretes oxytocin, or it could be from a cell that secretes ADH. And then there's other central nervous system disorders you don't need to know about yet. And the treatment with these people is to basically, you have to restrict water intake is the biggest one. You can give them ADH receptor antagonists, although this isn't very commonly used. The big thing here for SIADH is water restriction. So there's not a whole lot to the other posterior pituitary hormone, oxytocin. So really its secretion is triggered by mechanical stimulation of the nipple or mechanical stimulation of the cervix. This then goes to the hypothalamus and causes the release of oxytocin from the posterior pituitary in terms of the cervix. This will then, oxytocin being released will cause uterine contractions to help with delivery, and with breastfeeding, the mechanical stimulation on the nipple will cause oxytocin to be released. That will cause the breast to release the milk. And I believe that is all. And here, this may be tested on. I can't remember how much he likes to ask this necessarily, but basically giving oxytocin is what is used to induce labor. So now we have thyroid hormones. So the big things here is that the hypothalamus is going to secrete thyroid releasing hormone, which is going to act on the pituitary to secrete thyroid stimulating hormone, which then acts on the thyroid gland to stimulate thyroid hormones. So important takeaways from this slide is that TSH will go back and provide negative feedback to the hypothalamus to inhibit further TRH secretion. And then once you get to thyroid hormones, the thyroid hormones will act on thyroid hormone receptors in the pituitary and the hypothalamus to inhibit further secretion. So there's just feedback inhibition pathways for these two things, and that's sort of where they'll test that is at. So thyroid hormone works on both the pituitary and the hypothalamus, but TSH only acts on the hypothalamus in terms of inhibiting further secretion. All right, there's a couple of big points on this slide in terms of the things that's going to commonly show up. So thyroid hormone basically exists in two forms. We have T4 and T3. So T3 is much more metabolically active than T4. So when it's circulating around in our blood, we actually have a lot of T4. And then we have very little T3, but it's acted on by an enzyme that deiodinates over here and causes it to be more active. So it's one of the situations where we have a lot of this available, and then it can be acted on quickly if we need it. So T3 more active, less of it. T4 more abundant, less active. Then also the T3 hormone, or T4 hormone rather, exists on this thyroglobulin peptide, and it's you have the peptide here, and then these tyrosines are built off here, not built off this side chain here, and then this is the actual thyroid hormone, and these are tyrosine groups. That's really the big things to take away from this slide. So this is a pretty busy diagram, but overall a pretty good diagram, and he'll tend to pull questions from here in terms of what step happens next, or he'll describe a step and say, what's the name of this step. So here he's basically just listed these out in order that they occur over here. So you start off by having iodine trapping, oxidation, and then thyroid peroxidase, and then thyroglobulin. Then you have generation of thyroglobulin, iodination of the tyrosine residues on the thyroglobulin, conjugation, endocytosis, proteolysis, and secretion. So I'll just walk through this real quick. So the trapping is that TSH is going to open up this iodine and sodium symporter, so you're going to get iodine coming into the cell. And then you're going to have so this is on the sort of the luminal surface, and then this is the luminal surface, and this is the outer surface. And then on the luminal surface we have a port here, which I don't think at the time of this figure it was known, but it's not important for what we do. So the iodine comes in into the lumen, and it's acted on by thyroid peroxidase to generate negatively charged, or to change negatively charged iodine into inert iodine. And then these iodines will be added to the thyroglobulin. So it's important to also know sort of how these are added. So you'll get your two iodines on this one tyrosine group here first, and then you add on the second tyrosine molecule. So that's basically steps 3 and 4, and then step 5 is that you'll have some of this thyroglobulin vesicle will be endocytosed by the cells. Once we get inside the cells, you'll have this big thyroglobulin molecule that contains the T4 hormone, and inside of this vesicle, the thyroid hormones will be cleaved from this thyroglobulin, and they'll be recycled or broken down. Like they'll be broken down and then recycled. So then this vesicle contains just T4 and T3, and then that will be secreted. Just a couple of things that regularly show up from this slide. Is here these green boxes. So T3, if you remember from the signaling cascade, is one of the non-steroid hormones that acts on the directly on the nucleus. So that's something that will come up because it's kind of an oddball, and that's the way it works. And then here, this will be to test questions that can come from this material or from the signaling cascade. Is that TRH acts through the calcium IP3 DAG PKC pathway, and TSH acts through the adenyl cyclase cyclic AMP PKA pathway. And it's high likelihood that questions about which of those pathways each of these works through will show up. All right, so growth hormone. Typically, he's not asked too many questions from this, but I feel like he kind of has at some point. So it's important to know these nuclei in terms of the growth hormone. So from the arcuate nucleus, you're going to release growth hormone releasing hormone, which travels. It's released way up here in the stalk, at the superior end of the stalk, and then it travels down the hypophyseal portal system and into the anterior pituitary, where it acts on the somatotrophs. But the peptide that inhibits the secretion of growth hormone is actually secreted in the periventricular region, and that hormone is somatostatin, and then it inhibits the secretion of growth hormone by the somatotrophs. Sometimes this will kind of come up, and I think it's normally which nucleus it comes from it, so whether it's the periventricular or the arcuate nucleus. Again, so this just isn't stimulated for secretion. There's also negative action, which makes it somewhat unique. And then here, he had this question on our slide, and again, this was in the cell signaling cascade lecture. I told you to make sure that you know those examples, and that the growth hormone releasing hormone receptors, or cell signaling. I just talked about growth hormone, but be sure here to know that the growth hormone releasing hormone receptor actually acts through the G protein coupled receptors as well. As you can see here, we've got a receptor, a alpha beta gamma subunit, adenyl cyclase, cyclic AMP, PKA. So this is G-coupled protein, and it acts through the adenyl cyclase, cyclic AMP, PKA pathway. All right, some of this I've already said for growth hormone, but so up here we have the conditions that are going to activate growth hormone. So sleeping is good for you, activates growth hormone. Being low on sugar will activate growth hormone because it raises your blood glucose levels, and stress, aging, disease, and high levels of glucose will decrease it. So it comes from the anterior pituitary, and it acts on the skeletal system, the liver, the muscles, adipose tissue, which we've already talked about this. What we haven't talked about is that it also acts on the liver to induce the secretion of insulin-like growth factor 1. So this acts similar in the way for the way growth hormone acts, but it increases amino acid uptake, increases protein synthesis necessary for linear growth. So growth hormone secretion is stimulated by growth hormone releasing hormone, but it is inhibited by somatostatin. So somatostatin is in the hypothalamus and inhibits it up here in the anterior pituitary. Insulin-like growth factor 1 is a feedback inhibition. So we have here a final product that then goes back and acts on the hypothalamus to decrease the secretion of growth hormone releasing hormone. So that growth hormone acts in two ways. It acts by itself, and then it also acts through another hormone that it secretes. That's sort of the big points there for this slide. So this is a very important slide for this course, and it's really anything going forward. You're going to see this again in histo if you haven't already. You're going to see it again in phys. You're going to see it again in pharm. You're going to see it again in pathology. It's going to show up on step 1, etc. So you need to know that with the structure of the adrenal glands, we have the capsule, the cortex, which is this purple and blue part. You can see here it's divided into 3 zones, which we'll talk about in a second. And then the inner part is called the medulla. So there's a mnemonic for remembering this, and usually, the dirtier or more vulgar it is, the easier it'll stick. So you can think of outwards to bottom, you have capsule, then GFR, zona glomerulosa, zona fasciculata, zona reticularis, and the medulla. And then the mnemonic goes, the deeper you go, the sweeter it gets. So you have salt, sugar, sex. Aldosterone helps with reabsorption of salts. Cortisol increases blood sugars, and then of course you have the sex hormones that get made here. And then within the medulla you have epinephrine. All right, so for this slide, I included this one because, again, it's important for a lot of different things going forward. But the zone of the adrenal gland that these are formed in is very, very important. So everything starts up here, sort of in the outer cortex of the adrenal gland, and then they get fed down through the adrenal gland depending on which pathway you're going into. So here you have pregnenolone, which if you want to make aldosterone, it'll go through this pathway in the zona glomerulosa, and then you've got this series of enzymes that will lead to aldosterone. You'll need to know some of these steps later, but I don't think they'd be pretty low yield for Dr. Romero. And then if you need to produce cortisol, pregnenolone will then go to the 17 alpha hydroxy pregnenolone, which will then go to cortisol, and this again occurs all in the zona fasciculata. However, if you need to make testosterone, you have pregnenolone, and then you go into the zona fasciculata to make the 17 hydroxy pregnenolone, and then it'll be transported down to the zona reticularis, and then it goes down this pathway for testosterone. But this is really just to get at that these hormones are synthesized. There's a long synthesis pathway, and each of these synthesis pathways occur in each of these different zones, but they all start from the same precursor, cholesterol. So there's a different set of enzymes that are active in each of these zones, and that's why this occurs. And he's got this box here. I don't think I've seen this in a question yet, but it's probably important to know because he's got it in a box. All right, cortisol is our next anterior pituitary hormone. So this secretion is stimulated by some sort of stress response on the hypothalamus that sends signals to the hypothalamus, and so we've got diurnal rhythms, physical, emotional, biochemical stress. So this obviously isn't stress. This is just sleeping. But so they act on the paraventricular nucleus, which then secretes corticotropin releasing hormone, which is released into the hypophyseal system, travels down the pituitary stalk, goes into the anterior pituitary, which then acts on corticotropes. So again, kind of makes all these names are kind of linked to each other. So you have corticotropin releasing hormone, which acts on corticotropes. The corticotropes then secrete ACTH, and if you remember from the previous slide, there's initially a precursor protein called POMC, and then that POMC gets cleaved, and then that's where ACTH is contained. And then the ACTH then acts on the adrenal gland. And then you have feedback from the adrenal gland that inhibits this process. So definitely know these, again, from this lecture as well as the cell signaling lecture, that these are examples of these signaling pathways. So you can see here, corticotropin releasing hormone acts through the cyclic AMP protein kinase A pathway, as does ACTH. So this is a pretty busy pathway, and it's all interconnected. It's a little convoluted, but basically what happens is with aldosterone is that aldosterone is important in helping to maintain your blood pressure. So if you have decreased blood pressure, or if you have decreased blood pressure because you have low blood volume, this will help retain water through acting on the kidney to resorb salts. And I don't know if I mentioned this before in a previous review, but water tends to follow sodium pretty much everywhere it goes. So with ADH, it'll open aquaporins, which it's a water channel that allows water to pour in, but aldosterone is a little bit different in that it brings salt in, and then it brings in all the water molecules associated with salt. And what kind of shows up on tests a lot in boards and STEP and all that kind of thing is this box here, is that the RAS system, the renin-angiotensin system, is under a different set of controls from the aldosterone. Hold on, I got that mixed up. Because I'll just read it. Because mineralocorticoid synthesis primarily under the control of the renin-angiotensin system, hypothalamic pituitary damage does not significantly impact the capacity of the adrenal to synthesize aldosterone. So you'll see questions. Someone has blood pressure issues following a head injury or a tumor removal, and they'll almost always give you aldosterone or angiotensin as options to the answer to the question, because these help regulate and maintain blood pressure, but these act completely independently of ADH. ADH is not a part of the system. All right, so there's a lot of wonderful biochem, cell biology going on this slide, but really the sort of the take-home message here is that NED4 prefers to internalize and degrade ENaC. However, aldosterone comes into the cell, binds the mineralocorticoid receptor, acts on the nucleus, you get a transcriptional response, and it leads to the synthesis of this SGK1, which degrades NED4. So when you degrade NED4, this ENaC transporter cannot be internalized and degraded, so it stays on the surface. So this is how the transporter through which we reabsorb salt in response to aldosterone. From this slide, really sort of the biggest thing is just to know that cortisol is active, cortisone is inactive. I don't feel like he's gotten ever too nitty-gritty into the biochemistry of this, but cortisone inactive, cortisol active would be the highest yield things. All right, this slide is for the epinephrine. So what happens is you have a stress response, activates the hypothalamus, you secrete ACTH from the anterior pituitary, leads to release of cortisol from the adrenal cortex, which stimulates the synthesis of this PNMT enzyme here. And what this is important for is converting norepinephrine into epinephrine, and then that gets secreted out of the cells and increases blood glucose. The increase in blood glucose is because of glycogenolysis in the liver, lipolysis in the tissue. Remember, fatty acids are not gluconeogenic, but the glycerol backbone can be. And then glycogenolysis as well in the muscle. So this is important here. What he's got in bold is that stress increases both cortisol and epinephrine synthesis and secretion. So this is a busy slide I made just because he's not I don't think he had any slides where he kind of showed you what all these adrenergic receptors, what second messenger system they act through. So I just kind of just added this top part in here to just kind of show which ones they act through. So the alpha ones are GQ, which is the IP3 DAG calcium PKC pathway, and then alpha 2, and then all the beta receptors. Well, all the beta receptors are stimulatory, which are going to increase cyclic AMP, and then alpha 2 is going to be inhibitory, and it's going to decrease cyclic AMP. You'll go into more of this in Pharm, but he could ask questions again about, what's the example of a DAG IP3 one, and then an alpha 1 receptor could be an option. This is actually kind of low yield here, maybe useful later on. So really just focus up here on examples of these receptor types. So pheochromocytoma is an adrenal tumor that is going to cause excess catecholamine secretion, so norepinephrine, epinephrine. Really, and the cure for this is to remove it. What's going to happen is you're going to have patients that are going to have very paroxysmal, or what's the fancy word for episodic transient hypertension. So they're going to, at home, they're going to have they're gonna have hypertension, they're gonna be feeling like crap, they're gonna come into the office, you're gonna take their blood pressure and it's gonna be normal because it like ebbs and flows. And it's not constant. So sometimes it's kind of hard to catch. But yeah, it's kind of like sort of like having an anxiety attack, like constantly when you have one of these tumors. So you get the headaches, sweating, palpitations, anxiety. Like it's not good. So this is a disease that likes to show up a lot. All right, so steroid or sex hormone diseases. These are actually like fairly high yield in terms of like biochem and physiology and step one stuff. It's a long ways away for y'all, but never hurts to be looking forward a little bit. I had a hard time learning these. I kept getting them screwed up. You just got to just plug into it and commit them to memory. But so 5-alpha reductase deficiency is a deficiency here of converting testosterone to the dihydrotestosterone molecule, which is more active than testosterone. So if you have a deficiency, you're not able to generate the more active compound, but you still have some testosterone floating around. So what you get with 5-alpha reductase is you have they call minimal or absent adrenalization in utero. So they'll have typically very small genitalia. With androgen insensitivity syndrome, it's a defect in the androgen receptor. So the androgen receptor is going to affect testosterone and dihydrotestosterone. So you kind of lose all of your testosterone signaling. And you're basically going to end up with a female phenotype. You won't have ovaries. So you remember from developmental, the SRY gene this determines whether or not you're going to have ovaries or whether you're going to have testes. But because you're insensitive to testosterone, you have a female phenotype. You'll have regular breast development. You'll have female sex organs, but no uterus, and you won't have a whole lot of body hair. You'll also have the female psychosexual orientation. All right, big things from this slide. I'm not going to go through the whole the synthesis pathway for estrogen, all of this over here. You'll get that. You'll have to know that for phys. You don't really need to know it now. But cholesterol is the common precursor for all steroid hormones. Very, very important fact. And then here, you need to be able to make testosterone or you need to be able to make testosterone in order to make estrogen. So aromatase converts that to these estrogen components. I think the question will say something along the lines of, if you have aromatase deficiency or you don't have aromatase, then you can't make estrogen. Questions about oral contraceptives are pretty common, I believe, for Dr. Romero. So what happens is if you remember from the anterior pituitary axis, is that the anterior pituitary will secrete LH and FSH, which then acts on the ovarian follicle, which stimulates development and eventual release of the oocyte. And then once the oocyte's released, you get the corpus luteum, which helps maintain, which generates steroids or hormones to help maintain the placenta until it can sustain itself. So what you do, the whole purpose of the oral contraceptive pills that contain estrogen and progestin is that you're going to provide these hormones that these are going to secrete, and this suppresses LH and FSH secretion. Then these can't act on the follicle for development. And also, if you remember, the luteinizing hormone spike is what occurs to cause the oocyte to be released from the follicle. So you give estrogen and progestin, and then that you get feedback inhibition of LH and FSH in the anterior pituitary. So this does not occur. Actually, I may have gotten a little bit ahead of myself. So here's the menstrual cycle chart that I mentioned in the previous slide. So just for this, just kind of know what's going on in the follicular phase versus the luteal phase. So in the follicular phase, you have the follicular phase is predominated by FSH and estrogen, and then it's flipped in the luteal phase. The luteal phase is predominated by LH and progesterone. You have to have this LH surge here to get secretion or release of the oocyte, and It's probably the highest things here. Yeah. Again, the biggest things on this slide are in this box. Is you have the corpus luteum, which is the old follicle, which is going to it's first maintained by HCT produced by the embryo, and it secretes hormones. Then once the placenta is able to produce its own, the corpus luteum will involute and atrophy. And when you get to near term, human or HCG levels will drop, progesterone levels will fall, and then you have, which we've talked about, oxytocin already, but you also get this prostaglandin F2A, which this will show up for sure as well, which stimulate uterus contractions for delivery. Some of the stuff here could show up, it'd be kind of low yield, but knowing that at week 15 is around the time the corpus luteum involutes, yeah, that's about all I got. And then the pregnancy test this HCG level here. All right, so last slide, we're going to talk about prolactin, which is our last anterior pituitary hormone. So what happens is you have some mechanical stimulation of the breast, which then goes to the arcuate nucleus and the paraventricular nucleus. And what's going to happen is you're going to get inhibition of dopamine levels. You're going to get low levels of low levels. Dopamine levels are going to be decreased, which is going to stimulate the release of prolactin. And then also in this diagram, they show you what's going on with oxytocin as well, because it's a two-step process. You know, you have the mechanical stimulation to generate milk protein production via prolactin, but then you also have oxytocin, which is milk letdown. Some of this up here is a little esoteric and probably won't show up. So again, like I mentioned before, is that prolactin is, by default, it's inhibited by dopamine. So the stimulation here is to inhibit the inhibitor, lead to prolactin production. And then this here is something that shows up a lot too, is that prolactinoma is the most common type of pituitary tumor. And the question will be something like someone loses their bilateral vision. It's like they can't see periphery on either side because it's compressed in the optic chiasm. And it's the most common cause of that's going to be prolactinoma. So that's all I've got. Let me know if you have any questions, and I'll be happy to help out.